Okay, is my screen visible?
Not yet,
No,
sir.
sir.
Hmm. Let me know when it becomes visible, because from my end, it says it has been shared. It is presenting.
Sir, now it's been
Now
shared.
it's visible, sir.
Okay. 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 So I don't know when I got disconnected. So I was talking and then suddenly I realized there is no response. Then I realized that it has been disconnected. Okay. So the some of the important things that is required for cloning are DNA fragment, and this DNA fragment can come from a variety of sources. It could be, genomic DNA. It could be your cDNA, right? Typically, cDNAs are used for eukaryotic genes, right? It could be a plasmid DNA that somebody has given you so that you can use it for your own research. It could be a PCR product that you have, PCR, right? Or it could be a synthetic DNA. Synthetic DNA is something that we have modified and it has been synthesized commercially by a company. Okay. So the DNA insert must contain certain sequences at the end of the fragments so that it can be compatible with the vector in which we are going to clone. We are going to look at what a vector would mean. Okay. And so these two fragments, both the vector and the DNA insert can be joined together. We will see. I think we have seen earlier what enzymes are all used for for this joining together of two different molecules. Remember your classes during your basics of molecular biology when we talked about Okazaki fragments being joined together by DNA ligase, right? DNA polymerase keeps on replicating. So we are going to time and again hear some of those terminologies again and again. DNA ligase, right? Restriction enzymes, DNA polymerases. There are certain modifications to DNA polymerases, right? So these are called Klinov fragments. You will keep on listening to some of these things, okay, so that it gets etched onto your memory. Okay. So you need two things. One is your DNA fragment, which is DNA of interest, and this DNA of interest itself will be joined together to a vector, and this vector is generally a plasmid molecule. All of you know that plasmid is this extra chromosomal DNA that has this ability to replicate independent of the chromosomal DNA, right? So this is something that people have used it very successfully for cloning, right? Some typical, these extra chromosomal plasmids, they come in variety of shape, but we also have, by genetic engineering, we also engineered several plasmids. And so these plasmids are now given different terminologies. Okay, for example, cosmid. Cosmid is a large DNA vector which contains lambda phage DNA sequence, and it can carry fragments up to 45 kilobase pairs into the host. Typically, if you look at plasmids can carry on an average, very comfortably up to 2 kilobase pairs of DNA. Right? 2 kilobase pair means 2000 nucleotides. Right? Anything beyond that, generally the plasmids tend to become unstable. The plasmids will be lost, or the gene will be lost. So many complications happen. So typically, small plasmids, right? They typically carry up to 2 kilobase pairs of a foreign DNA. Okay, but there are certain specially engineered plasmids that can carry little larger DNA fragments, but generally they are not used in the routine molecular biology labs. Okay, cosmids are different from plasmids. Okay, and they typically can carry up to 45 kilobase pairs into a host. Okay, artificial chromosomes, just as we have normal chromosomes, people have created artificial chromosomes which can function very much like a chromosomal DNA in any organism. Okay. And plasmids, as we all know, is the small chromosomal circular DNA that can replicate in a cell. And this is what is commonly used in the for cloning purposes in routine molecular biology and biotechnology labs. Okay, so this is the structure of a plasmid that you're seeing. Okay, you have a promoter, that is going to drive this gene of interest that you have. Okay, and this gene of interest is cloned into a region called multiple cloning site. Some of this you may be aware. This is just reinforcing of your those concepts. Okay, and then followed by this, there could be a protein tag, or they may not be a protein tag. It doesn't matter, and it may contain a polyA signal. Okay, so this is typically polyA signal, protein tag. These are all used for your expression vectors. We'll come back and see what an expression vector would mean, what a cloning vector would mean. Okay. Hopefully in our, either in this class we'll start or we'll start in our next class. Okay. And then the plasmid will have what is called as an origin of replication. This is what makes the plasmid independent of your chromosomal DNA. So it can replicate independently. Okay. And then you have a selectable marker. These are typically antibiotic selection markers. It could be for ampicillin resistance. It could be for tetracycline resistance. It could be for chloramphenicol resistance. It could be for kanamycin resistance, right? There are several antibiotics for which you could select, but typically in a routine molecular biology lab, ampicillin resistance is the most commonly used one. Tetracycline is used on a relatively very infrequently, but it is nevertheless used. Okay, so this is the typical structure of a plasmid. It has a promoter that is going to drive your gene of interest, and this is called as the multiple cloning site. Okay, this multiple cloning site, if you look at the early plasmids, when the molecular biology revolution started, it used to have one or two restriction sites. Okay, multiple cloning site is nothing but a collection of restriction enzyme sites. Okay, and as things improved, now you have plasmids that carry several restriction enzyme sites, okay, in the multiple cloning site. And this is followed by a protein
Sir, your mic is on mute.
Sir, your mic is on mute.
he seems to be playing with this mic. I don't know who it is because I had started. Did it go mute in the middle, or when did it go mute?
In the middle, sir.
Okay, I don't know why. Okay. So if you look at this plasmid, you can introduce a plasmid both into E. coli, and you can also introduce a plasmid into either yeast or in mammalian cells, right? So these things are not essential when you introduce a plasmid into an E. coli protein tag or a poly-A signal. Okay, need not be there. Poly-A signal is definitely not necessary when you put it out in E. coli. But when you do this in yeast or when you do this in mammalian cell, poly signal is very helpful in terminating your this thing. Okay, and so this is a structure of a plasmid. Okay, so what are the elements in a plasmid? One is this origin of replication, from where the initiation of replication starts, right? And then you have a selectable marker. As I said, mostly these selectable markers are antibiotic resistance, right? But it could also be something else. For example, you can take an auxotrop, right? This is typically used in both bacterial and in yeast. You can take an auxotrop that cannot synthesize, for example, say a particular amino acids. It could be leucine, It could be histidine, or it could be, it may not be able to synthesize certain nucleotides, right? It could be uridine, right? So you could have those, this thing, and then you can have those particular genes here as a selectable marker. For example, you could put out a gene that is responsible for leucine synthesis, right? And so this can, when you transform an auxotrop, which cannot synthesize leucine by itself, when you put this plasmid and when you see which are now able to grow in a medium, not having leucine, because now that you have this particular gene which is responsible for leucine synthesis, you could use that as a selectable marker. But typically for routine molecular biology experiments, it is generally the antibiotics which are used as a selectable marker. Okay. Okay, so this just shows, what typically happens. Okay, so when you do this for bacterial selection, the bacterial selection would definitely have, as I said, an antibiotic resistance. So you will be introducing this into a bacteria, which is, to begin with, it will be sensitive to ampicillin. Okay, but whenever it takes up this plasmid, it will become resistant to ampicillin, because this particular one, the antibiotic resistance gene, it codes for certain enzyme that will degrade ampicillin. So by virtue of that, the E. coli becomes ampicillin resistance. Any bacteria that has not taken up this plasmid will remain sensitive to ampicillin. But any bacteria that has taken up this plasmid, will be able to grow in the presence of ampicillin. Okay. Okay, so this is typically the case. This is typically the case. So this bacterial cell has not taken up the plasmid DNA. So when you put it out on an LB agar plate with ampicillin, these cells do not grow. Okay. On the other hand, cells that have taken up this plasmid, when you grow them in medium containing ampicillin, they all multiply. They all multiply. You can see lots of colonies, right? So any questions thus so far?
No, sir.
Okay. So these are the typical properties of a plasmid. Let me just check. Let's just do one small quick exercise. Thought I could show you what this thing, how plasmid structure will look like. But I think we had seen this earlier, previously in our thing, last semester. Is my screen visible?
Yes, sir.
All right.
Yes, sir.
There we go. All right, we are back. Am I audible?
Yes.
sir. Yes, sir.
I think there is a lot of issues, net issues here at MIT. Seems to be very, very unstable. Okay, when this loads, maybe we will see. Okay, so once you make your gene of interest and your vector plasmid DNA, when you have joined them together by means of ligase. Okay, what does a ligase recreates the phosphodiester bonds? You remember your classes in basics of molecular biology. We looked at what does a 5' end means, what does a 3' end means. 5' end is always phosphate, 3' end is always hydroxyl. Phosphodiester bond is nothing but the formation of a bond between the 5' phosphate and the 3' hydroxyl group. This is essentially the function of a ligase which joins the 5' phosphate with the 3' hydroxyl group. Right. So once you have done that, you need to introduce this into bacterial cell, right? What is the bacterial cell that is typically used in the lab? It is generally E. coli, right? Escherichia coli, which is very commonly used for cloning, for doing protein expression studies and all that. Okay, the most popular one is E. coli, which is followed by Saccharomyces cerevisiae. If you look at the prokaryotes, then it is generally E. coli, and if you look at eukaryotes, it's generally Saccharomyces cerevisiae. Okay. Now we need to grow them in certain. You need to make these cells as competent. What does competent means? Competent means you do certain procedures by which these bacterial cells or Saccharomyces cerevisiae is able to take up this foreign DNA. So you are essentially incubating the bacterial cells with the DNA, and they need to take it up. They need to take it up so that it can now become part of the E. coli or Saccharomyces cerevisiae. Right. So competent cells are generally, they are made permeable to this extracellular DNA either by chemical means. You can, treat them with certain chemicals like calcium chloride, or you can introduce this foreign DNA by means of electroporation. That means you basically, give them electric shocks, very mild electric shocks for a very short time in the order of few microseconds. Okay, and typically, the cells have these pores opened up, and this foreign DNA enters into the cell. Okay, and then you are able to grow them up. Okay, so after you have introduced your foreign DNA into the cell, as we saw in the previous picture, some cells would have taken up the plasmid DNA, some cells would not have taken up the plasmid DNA. So we need to have some ways by which we are able to know which is the one that has taken up the plasmid DNA. So that's why I said initially when we start out with E. coli is the one that we are going to use it are generally sensitive to ampicillin, right? And so how many ever times you put a normal E. coli cell in ampicillin, it will be sensitive and it will never grow in ampicillin. But the moment you put this plasmid, okay, plasmids generally have ampicillin resistance marker, and those cells that have taken up this plasmid DNA will become resistant to ampicillin. Okay, so the selective medium is nothing but a medium that has antibiotics in it. Okay, as I said, the most commonly used antibiotics is ampicillin. All right, so back to the class. All right. Okay. So the selective medium generally will be a medium with an antibiotic like ampicillin, but sometimes you can also have either tetracycline, kanamycin, chloramphenicol. There are a few more antibiotics that we could use for selection, but most popular is ampicillin. Now let's see if we are able to see this. Oh dear. I can't believe that the page is still not loaded. Okay. If not, we will see in our next class. Okay. All right. Okay. Okay. We will look at some of the cloning methods. Okay. So there are several different approaches when we try to clone a gene. Okay. Depending on the research and the time available, people will choose different kinds of cloning methods, which is used to generate a recombinant DNA construct. What is a recombinant DNA construct? It is nothing but a plasmid which also carries a gene, which carries an extra foreign gene. Okay, so this is what is called as a recombinant DNA construct. Okay, most commonly used is the use of restriction enzymes. Okay, so restriction enzymes are the ones that cut DNA in very, very specific sequences, which are called as restriction sites. Okay, so you will have the same restriction site in both your plasmid DNA as well as in your DNA, the one that you're trying to clone it, right? For example, it could be EcoR1, it could be Hindi3, it could be any other restriction enzyme. You will cut it. You will cut your plasmid DNA with EcoR1. You will also cut your genomic DNA or cDNA or whatever DNA you're trying to ligate with EcoR1. Okay, and then you will incubate them together, and then you will add ligase to it. So typically, the ligase will join these two, okay, and then you will have the recombinant DNA construct. Okay. While setting up a PCR, right? PCR would entail the use of two primers, or sometimes people use more than two primers. So the primers can have your restriction enzyme site. It could be core1, it could be MH1, it could be any restriction enzyme site. You need to make sure that restriction site cuts you can not leave errors. Okay, so this could be done earlier. People used to do it with this thing experimentally, but now you have different ways by which you can do it in silico. You now have tools which are available which can tell you that this is going to get cut or not. Okay. All right. Okay. So this is typical example. The setup, right? And so this is your gene of interest. Okay. And then you have two primers which are going to recognize. We are going to carry a restriction enzyme site. So after you set up a PCR with these primers, you get a product. Okay, which will carry the restriction enzyme on either end. Okay, these restriction enzymes could be same or it could be different. Okay, for example, people can choose a combination of restriction enzymes. For example, you can have EcoR1 and Hindi3, EcoR1 and BAMH1, EcoR1 and any other enzyme. Okay, you can use a combination. And now this vector will also be cut with that particular restriction enzyme, whatever you're trying to do. In this case, there is a blue and yellow kind of restriction enzyme. This is just a schematic. So, these are represented again by blue and yellow, this thing. So when you cut, then you will have a and this thing. This product, PCR product, is also digested with restriction enzyme at the end of PCR. And so you actually have now, these are called as compatible ends. So this blue end will go and ligate only to the blue end. This end, the yellow end, will only go and ligate to the yellow end. So, these are called as compatible ends. Okay, it will go and The ligase will ligate these two ends. You now have a recombinant DNA molecule that can be transformed into E. coli or Saccharomyces cerevisiae, or it can be any other organism. Okay, so typically this step, up to the generation of recombinant DNA generally is the same. After that, the process by which this recombinant DNA gets introduced either into an E. coli or into a yeast or into the mammalian cell, the processes might be little different, okay, but not very different, not very, except that you can't be growing mammalian cells the way you're growing E. coli, right? So mammalian cells. All right. So I think the network issue, you know. Okay. Am I audible?
Yes, sir, you are.
Yes, sir.
Okay. Am I audible?
Yes, sir.
Yes, sir.
All right. I think there is some serious network issue at Amity, so you'll have to bear with me. Okay. All right. So that was, one way by which you could do is by restriction enzymes. Okay. The second is called as PCR cloning. Okay. Is my slide visible? It says presenting, but I'm not sure.
Yes,
Yes,
sir,
sir.
it's visible.
Okay.
Yes,
Okay.
please.
So when you do a PCR, PCR is typically done with polymerases like Taq polymerase, right? Most commonly. Okay. So when you do PCR with Taq polymerase, it has this unusual property of adding a single A nucleotide at the end of PCR product. Nobody knows why, but this is typically what is observed when you do a PCR with Taq polymerase. It adds a at the end of it. Okay, so typically what people have been, very smart in making sure that you use certain restriction enzymes, uh, use certain restriction enzymes so that they will have T overhangs. Okay, there are certain restriction enzymes, not all, but there are certain restriction enzymes. When you use those, you will have a T overhang. Otherwise, there are commercially these vectors are available. Okay, these are called as T-tailed vectors. Okay, all you need to do is set up your PCR. At the end of PCR, you need to mix these two. Okay, this T-tailed vector and your PCR product, and the ligase joins them, and you will have a recombinant DNA. See, either you use a restriction enzyme, or you could simply use if it is a PCR product, you could use a tailed vector. Okay. If it's not a PCR product, then obviously you cannot be using a T-tailed vector. Okay, so it depends on your, that's what I said in the beginning, when you, the method of cloning that you're going to choose depends on what the product is, how have you processed your products. Okay. Then what is, you have another thing called, so all these things, whether it's restriction enzyme or PCR, in this case we are using ligase for ligation, right? You also have certain other methods called as ligation independent cloning, okay, which is typically performed by generating short sequences at the end of a DNA insert to match these short sequences at the end of plasmid vector. Okay, and typically this would involve the use of certain enzymes called as nucleases. Okay, what are nucleases? Nucleases can be very broadly defined as enzymes that chew up the nucleic acids, right? Nucleic acids means it could be DNA, it could be RNA, it could be, any type of RNA. It could be tRNA, messenger RNA, or ribosomal RNA. Okay, so nucleases essentially are enzymes that have this ability to chew up the nucleic acids. Okay, nucleases again can be very broadly classified as exonucleases or endonucleases. Okay, what is the difference between an endonuclease and an exonuclease? Endonucleases would typically cut the DNA, okay, right in the middle, but they can't be starting at the end. For example, if you were to give a linear DNA fragment to an endonuclease, it will only start from the in the middle. It will never start from the end. On the other hand, exonucleases will start chewing up from this end and from this end. Okay, it can chew up from either end. So linear piece of DNA is chewed up by exonucleases. On the other hand, circular DNA is chewed up by endonucleases. Okay, that's the difference between exonuclease and an endonuclease. Okay. Now, these enzymes in ligation independent cloning, you use the exonucleases which chew up the 3' end, and they will generate a cohesive end between the DNA fragment and the plasmid vector. And these two can now, are combined, and this is typically becomes somewhat like an annealing step. Okay, and when you transform this mixture, the host ligase ligates these two products. Okay, so you're not using ligase from outside, but instead you're using the ligase from the organism to link these two things together. That's why this is called as ligase independent cloning. Typically for most cloning experiments, ligase is the most essential enzyme, but in this case, instead of you using ligase from outside, it takes the advantage of the organisms, this thing, and then it starts using the organisms ligase for ligase independent cloning. So this is what is schematically shown here. This is a PCR product, and when you use 3' 5' exonuclease, it basically cuts only at the one end. Okay, similarly you treat your vector with this ligase. Okay, and then when you join these two together, okay, you'll have this recombinant DNA. What is not shown here is there will be a small gap. Okay, because you have not ligated, there will be a small gap between the terminal nucleotide. Okay, but this gets sealed inside the E. coli or Saccharomyces cerevisiae. Okay. Then you have another one called as seamless cloning, which is very similar to the ligation independent cloning. Depends on matching the ends. Instead of using the 3' 5' exonuclease activity, you will be using what is called as the 5' 3' exonuclease activity, which will essentially create 3' overhangs, and the DNA polymerase fills in the gap and the DNA ligase seals the gap. Okay. There are minor advantages between difference between LIC, which is ligation independent cloning, and seamless cloning. Okay, different people use these things. Okay, but typically in a typical molecular biology lab, it is ligase. Ligase is the most common thing along with your plasmid vector. Okay, most people would use ligase for cloning. Okay, so this is schematically explained here. Okay, and you also have these days you also have what are called as different kinds of technologies. Different kinds of technologies have come, and you could use enzymes like topoisomerases for cloning. Typically, when people started thinking about cloning, ligase was fixed in their mind, but people also realized this topoisomerases have this ability to nick the DNA and then rejoin the DNA. Okay, so people started using the ligase topoisomerases for cloning experiments. Okay, another one is the use of site-specific DNA recombinases, which have this ability to recombine. Okay, and they have this ability to join two different DNA fragments. Okay, so this is what is now this is commercially available. Okay, as I said, the recombinant DNA technology has matured to such an extent that most of these are now commercially available. You don't need to do anything except that you may have to set up a PCR, and then you can get your vector of choice, and then you can start your cloning experiments. Okay, so this is sold by company I don't seem to recall. Okay, but let me. Okay, I'll probably give you the name of that company in my next class. Okay, but typically you have these vectors called as entry vectors. Okay, now you use your DNA and this entry vector along with recombinase enzyme, DNA recombinase enzyme. Recombinase enzymes are typically found in transposons, right? Transposons have this ability to jump around genes, right? Because they have an enzyme called recombinase. So you're using that recombinase, and that recombinase, what it does is it joins these two molecules. So you now have what is called as an entry clone. Okay. Now entry clone is nothing but a modified this thing, which is now carrying a piece of your DNA fragment. Okay. And then you would transform this into E. coli. Okay. Suppose you want to move from this entry clone to some other vector. For example, you may have cloned in a cloning vector, but now you want to express this gene. Okay. And this is expression vector is your destination vector. Then you can again use the recombinase enzyme to move your DNA fragment from this entry clone into destination vector, and it will get moved. Okay, all you need to do is incubate with the recombinase enzyme. So this is called as recombinational cloning. Okay, so there are different ways by which you could basically clone a gene, and we essentially saw few of the methodologies by which we can recombine the gene. Okay, so this okay, there we go. I just thought I should show you one of the this thing. Okay, now it won't move. Okay, so this is from from a commercial firm called New England Biolabs, which is one of the pioneers in producing restriction enzymes. Okay, and they also produce a lot of these commercially this thing used molecular biology tools. For example, they have many different kinds of vector DNA for cloning. There are many different kinds of vectors for expression. They have many different things. Okay, so this PUC19 is a commonly used cloning vector. Okay, and it carries the ampicillin resistance gene. This is a double-stranded circular DNA, which is around 2.6 kilo base pairs, or it has 2,686 base pairs, and it has a very high copy number inside the E. coli. Okay, inside the E. coli, this can be Okay, there we go. There we go. Finally. Okay, so this is how the PUC map looks like. Okay, this is how the PUC maps looks like, and this is the multiple cloning site. As you can see, it carries almost like more than 10 multiple enzymes, more than sites for more than 10 these things. Okay. Now these are advanced versions. These are advanced versions which carry even more restriction enzymes. Okay. So it depending on your need, you could use one of those vectors. Okay. So PUC 19 carries a 54 base multiple cloning site that contains about 13 different restriction endonuclease sites. Okay. So PUC, you have PUC 19, you have many other different kinds of vectors. These are called as cloning vectors. In our next class, we'll briefly look at what is a cloning vector and what is an expression vector. Okay. Um, any doubts?
No, sir, no doubts.
Okay. Okay. So if there are no doubts, then we will conclude our and we will meet tomorrow then. Tomorrow we have another class, right? So tomorrow we will meet and look at continue this discussion. Okay.
Okay, sir.
Thank
Okay,
you.
sir.
Thank you. God bless you all.
Okay, sir.
Thank you, sir.
Yeah. Yeah.
Thank you, sir.
Okay.
Thank you, sir.